Good afternoon, everyone. My name is Maddie Clingaman, and today I will be talking to you all about my research examining a microbial glycan degradation that I conduct under Dr. Higgins in the Department of Biological Sciences. So to start out, what is a glycan? A glycan is a large carbohydrate molecule made up of many small covalently linked sugar molecules. So this is a very broad definition, and thus glycans have very broad functionality within the human body, ranging from structural roles in cell membranes to recognizing viruses and immune responses to cell-to-cell communication and storage molecules such as starch or glycogen. So how do you degrade glycans? This is where glycoside hydrolases come, wherever glycans are within the body, so they're very broad. They play essential roles in breaking down carbohydrates, in modification of glycans, in biosynthesis, and catabolism of glycans, and so much more. So this research looks specifically at how microbes use glycoside hydrolases, or GHs, to break down glycans. So on this project, we're focusing on bifidobacteria as the microbe that we're looking at. And bifidobacteria is found in the gastrointestinal tract, and it seems to play a very important role in protecting infants from harmful diseases such as necrotizing enterocolitis, which is a major concern for preterm infants. So as you can see in the graphic, bifidobacteria is most dominant in breastfed infants as compared to formula-fed infants, and also elderly adults and kids. So what is it about breast milk that makes bifidobacteria so dominant? Although breast milk is mostly made up of water, there's also HMOs within breast milk, and these are human milk oligosaccharides. So these are glycans that the human body is incapable of digesting or incapable of breaking down, and this is where bifidobacteria comes into play. So when this was first discovered, there was a lot of questions about why the mother would produce these carbohydrates that the baby could not break down. And as it turns out, these carbohydrates within the breast milk help select for good bacteria such as bifidobacteria, like I mentioned earlier, which can help protect infants from diseases and different things to keep the infant safe. So within bifidobacteria, what breaks down glycans? So GH151 is a new fucosidase family of glycoside hydrolases. So this means that it cleaves fucose off of a glycan. So GH151 is the GH151 found in bifidobacteria, and it is known to remove fucose from glycans, like I mentioned. So the question is, can this enzyme cleave fucose from 2FL or 3FL, which are human milk oligosaccharides? And also, what else can BIGH151 digest? So the first step in this process is obtaining BIGH151 as a protein so we can study it. So BIGH151 occurs naturally. It occurs in bifidobacteria, like I mentioned. However, it's much more useful and easier to produce this protein recombinantly in the lab. And the way that we do this is by inserting a gene into a plasmid that can produce BIGH151, and then inserting this plasmid into E. coli cells. And we then plate and grow these E. coli cells in an incubator, and then they can be spun down into a pellet that we can then purify. And an advantage to recombinantly producing protein is the ability to engineer affinity tags on this protein, which can help with the purification process. So the first step in purifying the protein and making sure you have protein that you can work with is doing affinity chromatography. And essentially what this does is the affinity tag that I mentioned that's been engineered onto the protein binds to resin that's in a column, and everything else, all the contaminants such as E. coli and all of the other contaminants, flow through the column, and you're just essentially left with the protein of interest, in this case BIGH151. So once you have that, you have a relatively pure protein, but just to ensure that we have no other contaminants, we then do size exclusion chromatography, which separates the protein by size. And on the screen, you can see a size exclusion chromatography profile. And this large peak that you can see designates the fractions of interest of the protein the fractions of interest that contain the target protein that's been purified. So after you've done all of the purification, you're left with pure protein, and this is where you're starting to be able to work with the protein to do analysis on what exactly it's doing, and in this case, what exactly it's cleaving off. So we'd use TLC assays, thin layer chromatography, to measure the activity of BIGH151. And on the screen, you can see 4 different glycans or substrates that I've used with BIGH151. They're all boxed. The negative indicator shows just the substrate. There's no enzyme in there. And then the positive indicator is the enzyme and the protein together. And so as you can see on the first 3 substrates, the negative and the positive are different, showing that fucose has been cleaved off at some points during the first 3. And as you can see on the far right, there's just a standard fucose, so you can see that the 3 circled dots match where the fucose is, so you can tell that the fucose has been cleaved off of these first 3 substrates. However, on the 4th one, there was no activity. So this tells you the biochemical activity of this protein and tells you kind of what specific bond this protein can cleave. And then on to structural analysis. So your biochemical analysis tells you what the protein does, and your structural analysis tells you what the structure of the protein is and how the structure of this protein serves the function of this protein. So it kind of makes your biochemical analysis, it explains why your protein is able to cleave certain bonds and not others. So we use crystallography as a way to gather structural data. And crystals are formed by letting the protein crystallize in a variety of different conditions. And we currently have protein crystals and are now working on and we have preliminary structure and are working on getting some more structural data to get a better structure for our protein. So what are the overall findings and results? So as you could see on the previous slide with the TLC, BIGH151 is fully active on an alpha-1,4 glucanac bond, which is an alpha-1,4 linkage. And this alpha-1,4 linkage is also found in a glycan in the gut called Lewis A trisaccharide, which is also found in human milk and blood cells. However, when we tried the enzyme with Lewis A trisaccharide, there was no activity. And this could be due to the fact that Lewis A trisaccharide has an extra galactose. And then BIGH151 is partially active on the alpha-1,3 glucanac linkage. And 3FL, the HMO that I mentioned earlier, also has a similar linkage, but with a glucose instead of a glucanac. And when we tried the 3FL with the protein, there was no activity when tested. So this could show, again, that there's an extra galactose on 3FL, so that could be having some steric hindrance, or the fact that there's a glucanac instead of a glucose. And then future directions include determining a structure from crystal data and seeing why the structure is specific for the alpha-1,4 linkage. So thank you, and does anyone have any questions?
On slide 9, can you explain the different dots on the TLC and what they mean?
Yeah, so as you can see, the dots on the very bottom, so if you look at the negative for the very first negative, that is just the substrate. That is just the fucose alpha-1,4 galactose linkage. That's a substrate that we use to test. That's just the substrate by itself. And then next to it is where we added the protein in. And as you can see, you still have some of the original protein that didn't get cleaved and didn't fully get cleaved. But you also have a fucose and a galactose molecule or signal, and that shows where the protein had gone in and cleaved some of those bonds.
Why are 2FL and 3FL the only 2 HMOs that you tested?
So I mentioned that the GH151 family is a fucosidase family, which means it only cleaves fucose. And if you look at the structures of these different HMOs, 2FL and 3FL are the only ones that contain fucose. So BIGH151 would not be expected to have any activity on other HMOs.
Could you explain how you get good crystal structures?
Yeah, so we start out with doing a bunch of different crystal screens with hundreds of different conditions, with different buffers, with different salts, at different pHs. And once you do that, you see which ones crystallize the best under which conditions, and then you start to optimize those conditions using crystal trays. As you can see, there's a picture of a crystal tray on the screen. And you start to mess around with the different pHs of those conditions, different concentrations of the stuff that's within those conditions to see which one crystallizes the best. And then once you have great crystals, you can then send them into the synchrotron to get data that you can then use a computer program to solve to find a crystal structure and find the structure of the protein.